caught the research bug as an undergraduate at the University of Washington. I worked in heart and skeletal muscle mechanics with Dr. Mike Rainier and Don Martin. That's where I sort of switched gears from thinking more about medicine to really wanting to devote my career to research. And during my undergraduate, I did ROTC, so I took a little sojourn into the Air Force for a few years, and then came back into academia in a biomedical engineering PhD program at UC San Diego, and continued looking specifically at skeletal muscle with Dr. Richard Lieber. And we worked on collaboration with orthopedic surgeon, Dr. Henry Chambers, to look at kids with CP and how contractures form in their muscles. From there, I went to University of Pennsylvania to do a postdoc with Dr. Beth Barton. There we were looking at muscle hypertrophy and pathology, and got more and more interested in this idea of fibrosis and scar tissue in muscle, and how that impacts its ability to regenerate and function. I came to UC Davis about 2.5 years ago to start an independent research group. We call it the Myomatrix Lab. We focus particularly on muscle and its interactions with the extracellular matrix. So that's where the myo part from muscle and the matrix come together, and it's a lot more sort of interesting than Smith Lab, which is a little bit boring. But there's really sort of two main areas of our lab. One is that we're trying to use some advanced imaging modalities supplemented by biochemical techniques to really understand the nature of the extracellular matrix and the muscle tissue itself. What is the architecture, and how does that relate to the mechanical properties? We use some mouse models as well as biopsy tissue to measure those mechanical properties, and then try to relate those architectural features and see what's really driving, particularly in fibrotic cases, that excess of stiffness that limits the ability of patients to sort of move their joints freely in conditions of contracture. It's definitely something that we're very excited about. And one of the aspects that drew me to UC Davis was the ability to interface with the medical school here that does a lot of work with both Duchenne patients, Duchenne muscular dystrophy, as well as a collaboration with the Shriners Hospital for Children that sees a large population of kids with cerebral palsy. And so that's sort of harkening back to some of the work I did in graduate school, but we have actually recently just started a study to collect biopsy material from kids who are undergoing surgery for hamstring lengthening procedures, because their hamstrings are so tight, it doesn't allow them to ambulate very well. And so we're just now starting to look at those tissues using some of these second harmonic generation imaging, as well as some other biochemical techniques to really understand that. How is that architecture of the ECM changed in these subjects, and what aspects of that drive this stiffness? And we have plans, and we're actually working right now on isolating stem cells from those same biopsies so that we can probe their sort of innate abilities to regenerate and how that is modified by engineered substrates that we develop in lab. I was maybe sort of naively thought that the mechanics would just sort of scale with the amount of fibrotic material you had. So looking at sort of standard biochemical assays of like a hydroxyproline assay for collagen, one of the things we found is that was really actually a very poor predictor of the mechanics. And so the question is, what aspect of this matrix that we use some tools to show that there was a stiffening of the matrix itself, what other aspects of the matrix might be altered? And so in order to look at that and to understand, how is that, like the alignment of the collagen changed? For that, we need to, use these imaging modalities that allow us to see how that collagen is structured. So I'd say we're going more and more towards the imaging techniques that, for me, it's also just a little bit more rewarding to see some of these really cool pictures. I think they're quite beautiful to me anyway. But then to understand, there's a lot of information in those images, and try to piece together what aspects of that structure are dictating the mechanics. I'd say that's the main direction we're going, but we also are supplementing that with some biochemical data on perhaps cross-linking of the extracellular matrix as well, that also needs to be incorporated together, and really using all the tools that we have and integrating them to build a model of mechanics that is able to predict the passive stiffness in muscle. As a new lab, I sort of had my first graduate students join in just about a year ago, spring 2020, and that was right about the same time the lab shut down. So it made it a challenge to integrate sort of the building a lab and the culture, but we have certainly tried to make the most of the situation. In our lab meetings, we sort of incorporated an icebreaker question to start all of our lab meetings, just to allow the different people to get to know each other a little bit more. That doesn't happen when you're not in lab and the bench next to each other all the time. But it gave us a chance to step back and also dedicate some time to doing some more computational tasks that had sort of been hanging out there. We developed a machine learning algorithm to help us analyze muscle histology sections and calculate fiber sizes and fiber types more efficiently. One I alluded to a little bit is working more with human tissue, particularly in patients with CP, with cerebral palsy, that have these contractures, and really trying to understand what's going on in a human condition that doesn't really have an animal model that recapitulates the formation of these muscle contractures. Another area that I'm excited to pursue is further investigation of the mesenchymal stem cells that are present in muscle. We call them fibrodipogenic progenitors, that are really responsible, we think, for creating a lot of that fibrotic material. How does that fibrotic environment sort of feed back on itself in sort of a feed-forward mechanism on that population to really progress the fibrotic process? And are there ways that we can manipulate the matrix so that it doesn't have that feed-forward process within the FAPs? So another aspect of Davis, for those that haven't been here, it's fairly apparent once you get here, but it's a great town for biking. Everybody, one of the aspects, so it's completely flat. So take the kids out, lots of playgrounds, just sort of biking around, stopping off at a playground or having a picnic and playing with the kids. That's one of the more common things. Maybe stopping at a local brewery to grab a drink, but it's nice to enjoy the spring weather that way.
All right, we will finish with one last question. That is, how has the ORS supported you?
So the ORS, I think it's a really nice organization for me in terms of integrating more of the clinical aspects. So we really rely on working with orthopedic surgeons to access that biopsy material. And as I said, that's a exciting avenue forward for us that we'd like to incorporate more human studies. And so by interfacing with ORS and the researchers there that have that sort of more clinical experience than some of the basic science background that I come from, I think opening those avenues to those researchers and those clinicians who are interested in research is a really valuable part of ORS for me. And I think I was fortunate enough to be part of a research interest group this last year at ORS and trying to integrate more muscle into the program. And I think it's a key musculoskeletal tissue, and I'm hoping that we can help grow that within the ORS community as a muscle community as well.